Greetings and Assalamualaikum. The wonder of ATS beads trapping thiol group protein in various biological samples by Muhammad Arif Samshuri and Siti Nurtahirah Binti Jaafar. Protein is the most abundant component in biological system. Any changes of protein indicate the cellular response. Protein thiols are most sensitive to fluctuations in the cellular redox state and prone to oxidation. Upon physiological increase in oxidants, specific proteins could act as redox switches that regulate the conformation and activity of different proteins. Are radicals, or can be called oxidants, bad? The answer is yes and no. In small concentration, oxidant has an essential role in redox homeostasis. Redox homeostasis is essential for the maintenance of many cellular processes, including responses to oxidants, signaling, protection of protein thiols, oxidation reduction reactions, as well as the removal of xenobiotics. Otherwise, when in high concentration, oxidants can trigger the activation of transcription factor, or an FKB, while damage the mitochondrial membrane will release an apoptosis-inducing factor, or AIF, that activates protein caspases. This would lead to the death of the cells. Protein thiol can be found in all living organisms. Slight modification could give us insight on what happens in the cells. In addition, each of the modification is unique to each external changes. Image on the left shows the variety of oxidation on the thiol group of cysteine residue. Each of the modification can alter the biological processes. How ATS works in capturing thiol group in biological samples. Activated thiocephalose reacts with thiol-containing protein to form a mixed disulfide and release 2-thiopyridone. The solute is thus covalently linked to the thiol-containing protein, from which it can be subsequently eluted by addition of a reducing agent such as dithiotrietol or 2-mercaptoethanol. General methodology for ATS bead, which is developed from our findings, is very straightforward. From any biological samples, we must extract the protein by grinding the samples and calculate the protein concentration. If the protein concentration is low, we can precipitate the samples using ammonium acetate in methanol. Then, protein pellet is washed several times and resuspended in urea. ATS resin can be added and incubated for an hour to capture the protein thiol. After that, the ATS resin is washed several times using buffered solution. To release the thiol-containing protein, reducing agent is added and incubated for an hour. The ATS resin can be removed by centrifugation, and the supernatant containing thiol is removed and stored in minus 80 degrees Celsius for further analysis. This is the results of thiol-containing protein, where it was visualized by staining the protein with coma C blue. Different organisms and protein concentrations were used to present the effectiveness of ATS resin to capture thiol-containing protein. As for the cost of the chemicals, the total can reach 3,318 Malaysian ringgit without the labor cost. However, the real cost for one sample is 200 Malaysian ringgit, and if the additional sample procedures are required, such as sample cleanup and gel electrophoresis, the cost will exceed 200 Malaysian ringgit. However, we must note that some equipment used can be expensive for the first setup. Equipment used in this methodology is chiller centrifuge, while the consumables such as microcentrifuge tubes, pipettes and its tips are also required. Moving on to the concept of this methodology, the thiol-containing protein can be used in multi-purpose analysis such as in gel separation and protein sequencing. Then, the data can be used further to explore the differences among thiol-containing proteins in each biological samples. For example, if we found the biomarkers or novel cellular pathway in the sample, it can be used for designing further studies on the particular protein. Since we know that protein is the most abundant component in biological system, there are plenty of room for searching the novel protein. The uses of this methodology can be applied in control treatment experiment, environmental surveillance, and finally to elucidate cellular mechanisms. This is important in various fields of study, such as environmental monitoring. For example, protein that is known to be vital in particular organism can be used or targeted by using simple methods such as blotting, or we can develop rapid assay to produce results in no time. Another example for commercial potential is in medicine, such as development and effectiveness of drugs and biochemical pathways. As in conclusion, this methodology is simple and easily modified for any biological samples. Identifying proteins susceptible to oxidants is important to many fields of study. In medicine, it eventually helps increasing the well-being. Meanwhile, in environmental studies, it can be used as a tool for decision makers to include this fundamental component of life in their decision-making processes. There are few studies that has been successfully eliminating the use of ATS resin in proteomics studies, and hopefully with this straightforward methodology, many researchers can consider using this methodology in whichever field they are in.